PharmaPal.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
Today we're embarking on a really fascinating exploration into how labor begins.
Yeah, we're zeroing in on the pivotal role of prostaglandins.
Right, and also this intriguing idea about plant-based compounds maybe mimicking their actions.
Exactly. And especially for you tuning in with maybe a background in phytochemistry.
We think we've lined up some insights that might spark some real aha moments.
Definitely. We've got some compelling material to dig into. First, a big review. Global perspectives on elective induction of labor.
Okay, so a worldwide view.
Pretty much. It looks at practices and why induction happens globally. And second, we're sort of dissecting roles of prostaglandin receptors in female reproduction.
So getting into the nitty-gritty of how they work.
Exactly. It's a detailed review looking at the functions of these molecules and their receptors, especially around childbirth.
Okay, so our mission today seems pretty clear. Let's unpack how prostaglandins actually kick off labor.
Right, the molecular mechanisms.
And then explore this idea, exciting for phytochemists. I think that plants might have compounds doing similar things.
Connecting the dots there.
And finally, touch on the hormonal changes during pregnancy. How those shifts could, inspire finding new natural drugs.
Makes sense. A lot to cover.
Okay, let's jump in. Prostaglandins and labor induction. They really seem central to this whole process, don't they?
They absolutely are. The story, really picked up steam back in 1964.
Oh, that far back?
Yeah, with the discovery of prostaglandin F2 alpha, PGF2, and its notable effect on the uterus.
Okay.
That discovery really shone a light on how these lipid mediators could influence uterine contractions.
So fast forward to today.
Well, today, pharmacological labor induction often involves giving prostaglandins. Sometimes they're combined with, mechanical methods or even oxytocin.
In this practice, it's pretty common now. The Global Perspectives article suggested it's quite widespread.
That's a really significant point. Labor induction is a common procedure globally, but the rates, they vary a lot between regions.
How so?
Well, in high-income countries, you see pretty high usage. Like the US, it was around 23% back in 2010.
Wow, almost a quarter.
Yeah, and England, similar, about 22% in 2011-2012. Australia was up around 25% in 2010.
So it really highlights different approaches in those places.
Definitely.
What about in low to middle income countries, LMICs? Is it less common there?
Generally, yes. The data we have points to lower induction rates in LMICs, at least for births happening in hospitals or clinics.
Okay.
A big WHO survey across 22 LMICs found an average rate around 11%.
But you said generally, so there are exceptions.
There are, yeah. Quite notable ones, actually. Sri Lanka had a rate over 35%.
Over 35? That's huge.
It is. And Cuba was around 20%. So those figures are much closer to what you see in high-income countries.
Why might that be?
Well, it could reflect that the data from those specific regions maybe focuses more heavily on births that happen in facilities where induction is more likely.
That makes sense. Provides important context. Now, the reasons why someone might opt for induction, they're not always strictly medical, are they?
Absolutely not. I mean, there are clear medical reasons. Obviously, post-term pregnancy, past 41 weeks, membranes rupturing early, high blood pressure issues, stuff like that.
Right.
But elective induction before 41 weeks without one of those clear medical needs is becoming more and more common.
And why is that? The article mentioned a few things.
Yeah, the Global Perspectives paper talks about several factors, things like geographical remoteness, living far from the hospital.
Okay, wanting to be closer when it starts.
Exactly, or significant fatigue or discomfort for the mother. And sometimes just preferences around the timing of the birth.
So maybe trying to get a bit more control over when it all happens.
Precisely. The article also mentions that some doctors or midwives might have scheduling preferences or maybe even financial incentives, though.
No.
Well, the evidence supporting those advantages, especially for busy labor wards, seems pretty limited.
Interesting. And this also brings up the whole safety debate, doesn't it? Is inducing labor riskier than just waiting?
That is the critical question. And yeah, the article pointed out some conflicting evidence, especially about the risk of C-sections.
Right, I remember that. It wasn't clear-cut.
Not at all. The research findings are, well, a bit all over the place. some randomized controlled trials comparing elective induction to just waiting.
Yeah.
They surprisingly suggested a slightly lower risk of cesarean delivery with induction, but only in specific situations.
Lower. That seems counterintuitive.
It does. But then lots of observational studies comparing elective induction to spontaneous labor have suggested the opposite, a higher risk.
So why the difference?
It's really complex. Methodological issues like how you define the groups you're comparing or accounting for things like whether a woman has had babies before or how ready her cervix is, it makes getting definitive answers really tough.
Yeah.
So while some studies hint elective induction might lower C-section rates sometimes, the bigger picture is still complex. It really stresses the need for, careful patient selection and just more research.
Okay, so prostaglandins are used to induce labor. It's common.
Yeah.
But there's ongoing debate about elective use. Now, the big question for understanding the biology, how do they actually do it? How do they start labor on a molecular level? That second article, Roles of Prostaglandin Receptors, must get into that.
Right, and this is where it gets really fascinating. Mechanism-wise, both PGF2 and another one, PGE2, are known for their strong uterotonic effects.
Uterotonic, meaning uterus contracting.
Exactly. And the review highlights some really insightful studies using mice that were engineered to lack the receptor for PGF2 FP deficient mice, they're called.
Okay, mice without the PGF2 receptor. What happened?
Well, they couldn't go into labor.
Couldn't give birth at all. Wow. Why not?
The key thing was, in these mice, the levels of progesterone, the hormone crucial for keeping the pregnancy going, those levels didn't drop before birth like they normally do. It turns out that, in mice at least, that PGF2 signal binding to its FP receptor is absolutely critical for triggering parturition.
How? What does it trigger?
It basically tells the ovaries, stop making progesterone. This PGF2-FP signaling pathway, it kicks off a cascade involving GQ proteins that normally leads to the luteolysis, the breakdown of the structure that makes progesterone. Without the receptor, that signal is broken.
And when progesterone does fall normally?
That drop allows oxytocin receptors to increase in the uterus, which then primes the uterus, gets it ready for labor contractions.
So in mice, PGF2 is like flipping a switch from maintain pregnancy to initiate labor.
That's a good way to put it, yeah.
Yeah.
And the review also points to the COX-1 enzyme being important for making the PGF2 that causes this luteolysis and progesterone drop.
Okay, COX-1 makes the PGF2 for that part. But you mentioned PGE2 as well.
Right, because it's not just about progesterone withdrawal, it seems more multifaceted, even without that PGF2-FT signaling happening. Other prostaglandin receptors, probably EP receptors responding to PGE2, which is likely made by a different enzyme, COX-2.
Okay, EP receptors, PGE2, COX-2.
They seem to play a direct role in making the uterine muscle, the myometrium, contract.
So it's kind of a two-pronged attack. PGF2 helps get rid of the hold signal, progesterone, and then other prostaglandins directly stimulate the Kerr signal contractions.
That's a helpful way to think about it, yeah. They did experiments with mice that were FP deficient and had their ovaries removed mimicking the progesterone drop.
Right.
Even then, giving them NSAIDs, which block COX enzymes and thus prostaglandin production. That could still delay labor, but if they then gave those mice PGE2, it reversed the delay.
Showing PGE2 is involved in the contractions themselves.
Exactly. It strongly suggests PGE2 acting on EP receptors contributes to myometrial contraction. So it's the combined uterotonic actions of both PGF2 and PGE2 working through their different receptors that make them effective drugs for inducing labor.
This is really, really illuminating. And it immediately makes me think back to our listeners interested in phytochemistry.
Right.
If prostaglandins are so key, could there be plants out there with compounds that sort of copy these actions? The Global Perspectives article hinted at traditional remedies.
Yes, and this is where it gets particularly exciting from a phytochemist's perspective.
How so?
Well, that article mentioned easy-lumbezo from southern Africa. These are traditional herbal mixes used late in pregnancy to help labor along. And interestingly, when they analyzed these mixes, they found plant ergot alkaloids.
Ergot alkaloids. I know those have effects.
They do. They're known to have uterotonic properties. So historically, we have evidence right there of plants containing compounds that make the uterus contract, acting in a way that kind of parallels these labor-inducing drugs.
But the article also sounded a note of caution, didn't it? About risks.
A very significant caution that's critical to emphasize. Without proper standardization, consistent preparation and dosing and rigorous regulation, the safety and effectiveness of these traditional remedies can be really unpredictable. They could potentially pose risks to both the mother and the baby.
And this is where phytochemistry comes in.
Exactly. This is where that expertise is invaluable. Could we isolate specific compounds from plants, identify them, figure out if they could safely and effectively modulate these prostaglandin pathways we've been talking about?
Or maybe directly cause contractions, but in a controlled way.
Precisely. And for a phytochemist, knowing about these specific receptors, FP and EP, gives you targets. It tells you where you might look in the vast plant kingdom for compounds that could interact with them, maybe as agonists or antagonists.
It's a fantastic line of inquiry. So could we be searching for phytochemicals that literally bind to those EP or FP receptors?
Potentially.
Or maybe compounds that affect the COX enzymes, COX-1 or COX-2, that make the prostaglandins in the first place?
That's another plausible mechanism. It really opens up a whole range of research possibilities, doesn't it?
It really does. Okay, let's shift gears slightly to that other key piece, the hormonal changes during pregnancy itself. How could understanding those dynamic shifts be useful for, say, natural product drug discovery?
Right. We talked about progesterone being vital for maintaining pregnancy and how its decline in mice is a key labor trigger.
Yeah, but you also mentioned humans are a bit different there, with the placenta making progesterone.
Exactly. In humans, you don't always see that really clear drop in progesterone right before labor starts. The placenta just keeps pumping it out.
So it's not quite the same trigger as in mice.
Doesn't seem to be as straightforward. However, understanding the entire hormonal picture throughout pregnancy, the way levels of progesterone, estrogen, maybe other hormones rise and fall dynamically, that whole changing hormonal environment, the milieu, could still provide really valuable targets for natural products research.
So thinking like a phytochemist again, we could look for plants with compounds that maybe mimic or adjust the actions of these key hormones at specific times.
Exactly. For example, considering estrogen's role in making the uterus receptive for implantation, researchers might look for plant compounds that could subtly modulate estrogen receptor activity, perhaps in cases where implantation is failing.
Or maybe find compounds that interact directly with the hormone receptors themselves.
Precisely. Or even influence how those hormones are made or broken down in the body. This could really pave the way for potential natural product-based therapies for all sorts of pregnancy-related issues.
Like what?
Well, maybe supporting early pregnancy or managing preterm labor or other complications.
That sounds incredibly promising. The potential to find plant-derived compounds offering more targeted, maybe even safer options.
It's definitely exciting, but of course, and both our sources really hammer this home. Rigorous scientific investigation and thorough safety testing are absolutely crucial, non-negotiable.
Absolutely. Okay, so let's try and recap this deep dive. We've seen prostaglandins, especially PGF2 and PGE2, are absolutely central to starting labor.
Right. PGF2 is acting via FP receptors to lower progesterone in mice.
And both PGs contributing to uterine contractions via FP and EP receptors.
Check. We also touched on the history plants used traditionally, like those containing uterotonic alkaloids.
Highlighting both the potential there and the risks of using unstandardized remedies.
Definitely. And finally, we considered how those complex hormonal shifts during pregnancy offer potential avenues
for discovering new natural product drugs for reproductive health, thinking specifically from that phytochemistry angle.
You got it.
This has been incredibly insightful. It really leaves you wondering, doesn't it? With this complex dance between prostaglandins and hormones in pregnancy and labor, what amazing new plant compounds might still be out there waiting to be found? Could we find things that offer more targeted, maybe safer ways to manage labor or deal with other pregnancy issues, moving beyond just traditional use to really understanding how they work at a molecular level?
That is a really compelling question to leave you, our listener, with. And it's important, too, to think about the bigger picture.
Like the ethical side.
Yeah, and the global implications, especially in places where access to standard drugs might be limited. There's just this huge, fascinating landscape where phytochemistry meets reproductive health.
Holding potential for new solutions, grounded in understanding both plants and physiology.
Exactly. Lots to think about.
Thanks for tuning in to Bloom's Cast, brought to you by Blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
PharmaPal.